All right. Thank you, Tor and Nico, for setting up this amazing meeting. It's been really fun to participate in. I've already learned a lot. So I'm a psychiatrist in addition to being a neuroscientist, and one of the things we're really interested in my lab is whether we can take what we're learning about how the brain supports learning and memory and kind of motivated approach and avoidance behaviors and use that information to improve the way we go about diagnosing and treating different forms of mental illness. And as you can see from this slide, the take-home message is that we've talked about this already today. Many of these disorders, mental illnesses, are extremely costly, both in terms of disability and in terms of the amount of money we spend on treating them and problems related to them. And I think one of the reasons they're so difficult to treat well is something Tor and other people have mentioned today, which is that there's not a good mapping between our diagnoses in psychiatry and their biological substrates. And so it's hard to develop fundamentally new and more effective treatments. And I think one of the gaps there is a lack of objective biomarkers for diagnosing mental illness and subtypes of mental illness. So we've been trying to test whether we could use fMRI measures of functional connectivity to rethink the way we go about categorizing patients with depression in particular. There are many other folks who are really interested in this question as well. You already heard about Tor's work on a neurological signature of pain. It was this publication actually that was kind of inspiring to give this a try. And since then, a number of other labs have been trying related techniques for depression and other diagnoses. Carol Tominga has done really cool work with psychosis. Helen Mayberg on predicting response to DBS. Greg Siegel has used an approach really similar to ours for defining data-driven subtypes. So I think it's an exciting time to be working in this field, especially with the advent of these large-scale neuroimaging data sets that are being made available by the community, open to anyone who wants to access them. So I think there's a lot we can do in the near future. This problem of heterogeneity is especially salient in depression. We talked about this earlier today. I remember as a medical student, I think a lot of people share this experience, finding it puzzling the first time you learn about how depression is diagnosed. It seems very counterintuitive in some ways. DSM diagnoses depression when a patient presents with 5 or more of these 9 symptoms, meaning there's at least 256 ways you can meet these criteria, and that's not even accounting for the fact that some of the criteria are kind of, they contain opposites of themselves, like you can have weight loss or weight gain, you can be sleeping a lot or sleeping too little, you can be physically agitated or slowed, and no one really thinks that someone with weight loss, insomnia, and psychomotor agitation is likely to be suffering from the exact same biological problem as someone with weight gain, sleeping 18 hours a day, can't get out of bed, and is really slowed. But the fact remains that there's not really a better way yet of thinking about diagnosis in depression. Historically, the way we've gone about subtyping depression is by asking whether there are subsets of clinical symptoms that tend to co-occur in groups of patients, and then asking whether we can use those subgroups to identify neurophysiological, endocrine, biological, pick your favorite physical marker that might have a better correspondence between biology and clinical diagnosis. And we've learned a lot from these efforts, right? I think like some of them, like seasonal depression is one that has seeped into the public lexicon. Everyone's familiar with it. But the fact remains that there's really still not a one-to-one correspondence between almost any biological measure and a diagnosis of depression that's useful in a like widespread clinical context. So another way to think about this problem is to kind of turn that on its head, ask whether we can subgroup people on the basis of biological or other signatures, and then if so, then we can ask, do those subgroups predict different clinical symptom profiles, different prognostic outcomes, and perhaps provide a better mapping to biology? And that's what we set out to do. The work I'm going to present to you was made possible by the folks you see pictured here. I like to highlight them at the beginning because they really are responsible for making this work happen. Many of these people shared their data with me. I went around the country basically begging, and many of them shared their data with me before it was even published. And I think it's this kind of spirit of openness and collaboration that's really promising for the future. A lot of people are coming around to the idea that we can do more by working together than we can by working by ourselves in each of our labs. So we initially acquired this fairly large data set for depression imaging, 711 subjects, 333 unipolar depressed patients that were acquired from 5 sites. And we used it to try to discover subgroups of patients, to train classifiers for diagnosing them, and to learn a little bit about how the subgroups differed. And then critically, later, additional data became available to us, and we could use that to replicate the results we found in the first data set. So just looking at the time, I'm going to be presenting some of this work again on Thursday morning. In the spirit of the theme of the symposium today, I'm going to focus more today on some of the methods and what we learned, stuff that we don't get an opportunity to talk about very often. And I think I'll try to emphasize things that might be tips that might be useful for people who are interested in applying similar approaches to related questions, particularly in clinical populations. That might mean jumping over some of the actual findings, and I'm happy to address any of those at the end in the question session if people are interested. The first thing we set out to think about and address was how do we go about controlling for artifacts, in particular site-related differences that might confound the results we get from integrating data across many sites. And I always emphasize we're not, in my lab, we're not really driving these kind of methods forward, especially with respect to correcting for motion-related artifacts. Rather, we try to be up to date on the literature and implement kind of best practices. And you've already heard today from some of the people who are kind of pioneers in this area. For people who aren't familiar with neuroimaging data, I also work with a lot of wet lab-based scientists. I like to show these images because I think it really makes it clear what a big effect motion can have. So what you see on the left is a mean kind of functional connectivity map for the whole brain. Warm colors denote positively correlated areas, cool colors, anti-correlated areas. And then you see individual subjects that are kind of typical in terms of their motion, and at the far right, individual high motion subjects. And you can just see anyone with no experience can glance at this and see what a large effect motion can have on these correlations. You see in the middle this kind of there's structure to this correlation matrix, and on the right, a lot of that structure seems to be lost, and overall, warm colors, high correlations tend to dominate strongly, and a lot of people have seen that. You can see that also by, and this is a theme we've talked about multiple times today. I'm just going to underscore it. I'd really encourage everyone who's maybe new to this field to really take some time to manually, visually inspect all of the data that you want to analyze. What you see here are just histograms of the correlation coefficients between regions of interest spanning the entire brain. In blue, you see the probability distribution for all subjects in a healthy control data set, and in orange, you see individual subjects superimposed on them, and the thing you can see really quickly just by glancing at it is that most of them look pretty similar to the overall mean for controls, but that is not the case every now and then for a high motion subject. The whole distribution is strongly shifted to the right. So we need to think about how to control for these kind of artifacts. The way we went about doing it was just by trying to implement what other people have done kind of at the more stringent end of the spectrum of how you might go about controlling for motion-related artifact. We censor high motion volumes. We define those as volumes where the frame-wise displacement exceeds 0.3 millimeters. We perform simultaneous nuisance signal regression and bandpass filtering to avoid the effects that some people have observed where bandpass filtering prior to nuisance signal regression can kind of spread out the motion artifact into volumes that don't get censored based on that frame-wise displacement threshold. And then we end up excluding subjects altogether if the amount of data that is remaining after censoring is less than about 4 minutes in our data. I think there's good data out there to suggest that even a little longer would probably be good for robust detection of these low-frequency oscillations and correlations across brain regions. As always, whenever you're trying to implement kind of data quality controls, there's a trade-off, which I'd encourage everyone when you're looking at this kind of data to bear in mind and think about how you want to implement that trade-off. There's a trade-off between stringent data quality and data preservation. And I'm going to show you how that played out in our data for a couple of minutes here. After controlling or attempting to control for the effects of motion, we tried to test, and we also published in the supplemental figures, how well we were able to actually control for those differences. You see in panel A the distribution of frame-wise displacement across volumes in patients and controls. They're very similar, so we don't think that frame-wise displacement, that head motion differences between patients and controls were contributing to the results that we saw. We could also ask how well different methods for controlling for motion-related artifact, how well they actually accomplish that goal. These plots are really directly inspired by the work of Jonathan Power and other folks looking at how functional connectivity features differ in high motion versus low motion data. That's what you see in panel D at the bottom for 3 different ways on the right, no correction for motion artifact. In the middle, the ANAT-ICOR method plus a 0.5 millimeter sensor, and on the right, ANAT-ICOR plus 0.3 millimeters. And you see that we can do a pretty good job of reducing the number of functional connectivity features that we see coming up as different in high motion versus low motion subjects. In panel E and F, you can see how we go about thinking about like what the right threshold to select should be. You can see in the upper right how the number of connectivity features that differed in low and high motion subjects varied as a function of the threshold that we used. As you'd imagine, that goes up with more liberal thresholds, and it really goes up sharply, like around 0.5 in our data. And then there's also on the bottom right panel F, an illustration of the trade-off of stringency in correction and data preservation. Below 0.3, or really like 0.25, there's a pretty steep increase in subjects that we had to exclude altogether. I think there's probably significant promise for further improvements upon the way we did things. The data we were working with, some of it was collected, almost 10 years ago. I think now that people are much more aware of how important it is to not have your subjects moving their heads during the scan, the head motion tends to be reduced. So I think that could be improved upon even further. I think the main thing I'd like to emphasize here from a training perspective is just I think the field would really benefit from more transparency in reporting on the way we go about correcting for motion and on what motion looked like in the data that we're using and how it affected the data, just for the sake of making it easier for folks to further improve upon things in the future. Site-related differences are also a big concern, particularly when you're dealing with neuropsychiatric, like clinical populations. We found that in some sites that the data could vary systematically in terms of their head motion by site. We don't know why. That might have to do with how the investigators went about habituating their subjects to the scanner, training them not to move their heads, maybe related to subject comfort for whatever reason in the scanner, temperature of the room, their position of their head in the head coil, who knows, but some sites had higher motion than others. Clinical heterogeneity is another concern, maybe less so when you're not dealing with clinical populations, but if you're trying to discover subtypes of an illness and there are like clinical demographic variables that differ in a way that's collinear with site-related differences in functional connectivity measures, that's going to be a real problem, right? So you want to be mindful of that issue and think about how to control for it. Recruitment criteria can certainly contribute to that. Medication usage and differences across site can contribute to that as well. And then another factor that we and others have seen and that you should be mindful of are regional differences in MR signal quality that can differ across sites depending on the type of head coil that you use, the pulse sequence you're using, the scanner. So you want to make sure that, again, that those differences aren't collinear with site in the data that you're trying to discover subtypes in. Here's kind of an overview of our methods. A lot of this work was led by a really talented MD PhD student, Andrew Drysdale, who's now a psychiatry resident, and Mark Dubin, my colleague at Cornell, is the single largest contributor of data to this work. So having attempted to control for motion-related artifacts and site-related artifacts, we first asked whether there were connectivity features in the kind of the whole brain architecture of functional connectivity that correlated with specific symptom combinations. So this was basically a feature selection step, right? We wanted to cluster patients on the basis of a subset of connectivity features that might be relevant for depression, and this was one way to go about it. You could imagine other ways. And then using those features, we tested whether they tended to cluster into patient subgroups. If they did, could we optimize classifiers for diagnosing these putative subtypes and differentiating them from healthy controls? Then we could test for clinical differences and try to replicate them in an independent set. And that's what I'm going to describe in the last few minutes here, again with an emphasis on some of the tips that we learned along the way methodologically. I'm going to skip over this because it's probably familiar to most of the people in this room. We tested different methods for parcellation. The results I'm going to describe today used Jonathan Power and Steve Peterson's functional parcellation of the brain into actually 277 functional ROIs, 258 of which had strong signal coverage across all the sites in our discovery sample. And we used canonical correlation analysis for dimensionality reduction. So we searched for features that were predictive of clinical symptoms, and then we developed a low-dimensional representation of those connectivity features that were predictive of particular clinical symptom combinations in a data-driven way. That's what CCA is doing, kind of analogous to PCA for folks who aren't familiar with it. We discovered two components, which we term, for lack of a better name, anhedonia-related and anxiety-related connectivity. We performed hierarchical clustering to ask whether patients could be clustered into subgroups along these two dimensions. We found that they could. This is another aspect that I don't talk about often in most talks, but I think it's valuable methodologically if people are interested in trying this in their own data. You want to make sure that you're not clustering on all those artifacts that I just described. So one of them is motion. Can we convince ourselves that that the subtypes aren't just driven by differences in motion? We found that motion wasn't different across the subtypes or between patients and controls, and that even when we excluded any connectivity features that differed between high and low motion subjects, the cluster identity was almost always the same. And you can see on panels I and J that the percent of subjects who were correctly diagnosed based on functional connectivity features in black versus functional connectivity features plus motion in gray were not different. So we don't think that motion is contributing meaningful information to how these subjects are clustered. They also didn't differ by medication status. I think that's something that's definitely worth examining if you have patients who are medicated, because we do know, I'll emphasize, although our clusters didn't differ by medication status, we know from lots of work that medication usage can have effects on functional connectivity and does have effects. You could also imagine different clustering solutions. So I thought it might be useful to walk you through our thinking on how to go about evaluating and comparing different clustering solutions. One way is to ask about the quality of the clustering solution. How well does a given clustering solution parse variance in your subjects? Kind of like this variance ratio criterion, kind of like an F-test, is the variance between groups larger or smaller than the variance within groups. And you can see that this criterion was maximized at our 4-cluster solution, but you can also see by looking at the axes that the differences were modest. So I don't think this alone would necessarily dictate what should be the optimal clustering solution, but it's one useful piece of information. The other factor we considered was a more pragmatic one, which is a clustering solution in the data that we have is only useful to us if we can learn something from that clustering solution, and that's going to be limited by the size of our sample. And so if like a 5th or a 6th cluster results in a sample size within that cluster of 5 or 6 people, that's probably not a useful clustering solution to us, although it may be biologically or clinically interesting. We just can't learn anything about it with such a tiny sample in the data that we have. So we looked at how different clustering solutions varied in terms of our statistical power to test for differences and in terms of cluster size. And here the drop-off was rather more steep after 4 clusters. And that's partly why we focused on this 4-cluster solution. I always emphasize in these kinds of talks that I don't think that the solution that we're describing here is the only solution or probably even the best solution. If we had more data available to us, richer phenotyping of the patients, we might be able to learn. We probably could learn a lot more from the sample. But what I want to try to convince you of this is one useful solution and hopefully a framework that can be improved upon. I'm going to skip over this part in the interest of time, but I would also, for people who are interested in this kind of clustering analysis, just encourage you to think about the stability and robustness of the clustering solution. If it's real, presumably when you cluster across many different subsamples of your data, you should get relatively similar clustering results. And so I'd encourage you to examine that. And that may also be a useful factor in evaluating and comparing different clustering solutions. Okay, I'm going to take the last 5 minutes to tell you a little bit about some of our findings. I'm going to skip this part, basically just suffice it to say that we looked at the relationship between functional connectivity in our 4 subtypes and clinical symptoms. And to summarize, we found basically that there were some functional connectivity features that were shared across all 4 subtypes, and that they were predictive of the severity of clinical symptoms that were also shared across all 4 subtypes, including depressed mood and anhedonia. These 2 symptoms, one of them, you have to have one of these symptoms to have a diagnosis of depression, so it makes sense that almost all patients in our sample would have them to some degree. On the other hand, there were also functional connectivity measures that showed strong differences across subtypes. That's what you see here, kind of like a fingerprint of each of those 4 subtypes. And there were features that differed, and they were predictive of differences in clinical symptom profiles in a way that was kind of intuitively appealing and maybe useful for generating hypotheses for testing in a future data set, but we're really not able, with the data we have here, to tell you anything about how particular connectivity features might relate to particular clinical symptoms. Having discovered these subtypes in a subset of the data that was kind of rigorously controlled, such that it came from just 2 sites with identical scanner protocols, identical scanners, identical inclusion and exclusion criteria, very similar medication and diagnostic breakdowns. We then trained classifiers to diagnose the subtypes. Again, I don't think it's so important here, but we were able to significantly improve the performance of these classifiers, such that the best performing classifiers in leave one out cross-validation gave us accurate classification rates in the range of 84 to 90% approximately. And we were able to ask whether the subtype diagnoses were stable over time. That part, I think, is really critical. And when you're thinking about designing future studies, if you're trying to do something like this, I'd really encourage you to try to get 2 separate scans from at least a subset of your subjects, so that you can ask whether a patient diagnosed with a given subtype today gets diagnosed with the same subtype in a month. I think that's really important for understanding whether you're subtyping on something biologically and clinically meaningful, or is it like what the patient happens to be thinking about in the scanner at the time of the scan, whether he had a fight with his spouse prior to the study. Factors that could influence functional connectivity, but that might not be interesting to us biologically. I'll also just point out that those results aren't a foregone conclusion. So if you cluster on clinical symptoms, you get very unstable clustering solutions, and you get very unstable, longitudinally unstable subtype diagnoses. Having that second data set afforded us the luxury of being able to replicate it. Like Tor described, like you'd expect, the performance of our classifiers was a bit lower in the replication sample, but it was still respectable. The worst performing ones were around 80%. And we could also, I'm actually just going to skip this part, and please come on Thursday morning if you're interested in hearing about how they cross diagnostic boundaries. And in the last couple of minutes, I'll just tell you about how they predict treatment response. So we had found that one of the findings that stood out in the functional connectivity results was that the subtypes differed strongly in their dorsomedial prefrontal cortex functional connectivity. And we know that, this therapeutic intervention, transcranial magnetic stimulation, that although dorsomedial prefrontal isn't the conventional FDA-approved target, we know that dorsomedial prefrontal cortex is effective as a target. Multiple people have showed that in fairly large samples. And we know that from our work, from Jonathan Downar's work, from Amit Etkin's work, other folks, that your functional connectivity at baseline is correlated with your likelihood of responding to the treatment subsequently. So we just asked, based on the finding that dorsomedial prefrontal connectivity varied strongly by subtype, whether the subtypes differed in terms of their treatment responsiveness. And to summarize a kind of busy slide here, we found that they did. Subtypes 1 and 3 were more likely to respond to TMS targeting this node than subtypes 2 and 4. And we could train classifiers to predict who would go on to respond to TMS based on their functional connectivity prior to treatment. And we could do significantly better in panel G if we included their subtype diagnosis in the classification than if we just had connectivity features alone. I also told you that their clinical symptoms differed by subtype, so we wanted to answer whether we could use clinical symptoms alone to predict whether they would respond or not. That could be really useful and cheap and powerful, because this is a treatment that takes 5 weeks to take effect, so it would be great to know in advance. And to that end, we just tested whether clinical symptoms were predictive. We found that this was a statistically significant result. We could predict who'd go on to respond, but not a clinically meaningful one. 61 or 64% isn't so much better than chance, and probably not worth the cost in this context. We also tested the connectivity and subtype classifiers in an independent replication sample, and got, again, fairly similar results. So I think that's important. I always like to be thinking about how to validate the results when we're thinking about data-driven subtyping. Tor mentioned no one needs a fMRI scan to make a diagnosis of depression, but an fMRI scan could be really useful if you can do something that you can't do just by interviewing a patient. And one of those things is predicting who will respond and who won't, and mapping particular treatments to particular patients. And I think that this approach holds some promise in that regard. So with that, I'll stop. I'll summarize some of the main points. I think it's important to think about managing the competing demands of sample size versus data quality, and examining and considering how clinical heterogeneity, medication status, and head motion differences across site might be influencing the results you observe, and also being kind of as transparent as possible and reporting as much as you can on how you go about implementing these methods, and mindful of the limitations not just of our neuroimaging data, but also of our clinical scales. And lastly, large samples are useful. We can do a lot, I think, with bigger data sets than we can with very small ones. So just be mindful, I think, of the limitations of the data that you have at hand and how best to use them. And with that, I'll stop. I just want to highlight again the folks in red who made this work possible by sharing their data with me, and my sources of financial support.
Come up here. So functional connectivity is really useful when you have resting state data and you don't know the alignment with respect to time of your data, right? However, I'm wondering, might it be even more powerful at some point if we had paradigms where we probe the brain with stimuli, like some of the stimuli that Tor showed, and this would put us in a situation where we have this temporal alignment, and then we can look at many other summary statistics, or just directly at the patterns of activation across the brains in order to get biomarkers for different disorders.
Yes, I agree completely. That's a big limitation of the data we had at hand. That's one of the things we want to do going forward to kind of, I guess, version 2.0 of this project. I think in addition to kind of psychophysical stimuli that could constrain your analysis of the data, some of the neuromodulatory techniques that we heard about earlier in the day, TMS, maybe, TDCS methods like that could also be really useful. There's data out there to suggest that different patients, that individual differences in the way the brain responds to these kind of stimulation interventions acutely may be predictive of interesting clinical outcomes as well. So that's something we want to try too.
Thanks.
Hi, thanks. Yes, my first question is about the TMS study. Did you publish that already, or?
Yeah, the TMS data that I just showed you is the last figure of that same paper.
Okay, so and the second, it's more of a comment about the work. What do you think is the trade-off between getting less motion artifacts and the sample size? You mentioned a little bit about it, but it seems to be a big thing, and we're trying to attempt something similar with the TMS as a treatment for cocaine addiction in our case, with a big data set first of 100 and something subjects, and then trying to see it predict who's going to benefit from it. Can you comment on that? Because there's a lot of, we're having a lot of drop-offs because of the motion. And what do you think is the good trade-off between that? Because getting more subjects means also more money. And
Yes. And yeah, like large data sets are good. The way I think about it is I think there's a pretty clear-cut, like black and white recommendation on like high motion subjects. Sometimes, like I showed you at the beginning, have that whole like giant shift. That data, I think, is clearly probably garbage data, and having that in your sample is not going to be useful. Where it gets trickier is like we find that almost all subjects whose connectivity histograms look like that are high motion subjects, but we don't find that all high motion subjects have data that looks like that, and I don't know why that is. And so that's where I think maybe in a subsample of your data, some data-driven exploration of the trade-off between censoring and data preservation can be useful. My take, again, I'm not an expert in this area, but my take is that there isn't yet, like, there are some questions where we have pretty well-defined answers, but I don't think there's like a black and white recommendation of do this is the way to do it. But try to be thoughtful about it and transparent about the outcome. I think that's the best advice.
Thanks.
Yep.
Now we have a packed panel. More painful fear. All right, let us have it. If you guys don't grill us with extremely difficult questions, we're going to have to start asking each other embarrassingly difficult questions.
Well, I think he had a question for you, Tor.
Oh, yeah.
There's one.
I have a question more for Tor, what you presented. I really like your toothbrush analogy. And I'm trying to figure out how that would correspond to, so it's develop, replicate, reproduce, replicate, generalize, or something along that lines. The first step is develop, and it seems like we're stuck in that phase because of the toothbrush paradigm. So everyone wants to create their own things, and it's very difficult to have replication studies. It's becoming less so, but replication studies to be published. So what's your thoughts on that?
Well, right. A couple different things, I guess. And one thing just to clarify is that I feel like the idea of sharing a model is not something we typically think of, right? We think of sharing data or even sharing results or maps. So I just think that's kind of an important key thing, and it is kind of like a resource, right? And I think part of the problem is that everyone sort of wants to create that, maybe be the creator of that model or something like that, you know? Now, in terms of your specific question, in terms of replication, one hope is that if you have something, okay, so let's take Conor's work, right, and colleagues, and so that's in a sense they did several steps of like, development and validation and so on. But then it would be really helpful if another independent group said, well, we got data from depressed patients too, right? And apply the exact same model, maybe exact replication, maybe conceptual replication. That's not going to solve the sociology problem if people are less interested in sort of replications. But what can happen, though, is that you can then build off of that model. So that's a platform for saying, oh, well, gosh, we have a new treatment. We have focused ultrasound for depression, right? Like Gary talked about this morning or didn't talk about. And then, now let's see if that affects, is it work for only one of the biotypes? What effect does it have? Does it affect those particular patterns, et cetera? So then you end up with a replication that's embedded in a new test of something else. And so then that's really, to me, that's cumulative science, right, which is super exciting.
So the Sixth Sense data that you presented, which was published and had some press, was not replicated in other studies, of course, and tried to submit to the same journal, who said, we don't publish replication studies. So there's this obstacle, not only among researchers, but also among, well, I guess that's the journal saying that. It's probably not the peer review process. Well, I don't know who said that. So
I just have to say, the Nature journals have written a ton about replication crisis, and there's a bunch of Nature editors here, so we should all tell them.
There's also, this year, there's the first time there's the OHBM Replication Award. And I was very heartened to see that it got, I believe it was 27 submissions, which I think is fantastic. So I think we're moving in the right direction.
Question in the back?
So I had a question about MVPA. So I was wondering, it seems to me sometimes like there are some labs that kind of do MVPA and there are other labs which tend to not do MVPA. They do sort of the more traditional analyses. But I was wondering if you have any sort of advice for, like, let's say you're starting a new task. How do you decide which way you want to analyze it, or if you have any general thoughts about that?
I do, actually, and this will let me give Tor a hard time about what we were just talking about, too. My thought is that don't worry about it. You start with the science, and you ask your question. You say, this is what I want to learn, or like, this is the mystery that I want to solve. And then you think hard about what's the best analytic approach that I can use to solve it. The analytics are just tools. And, as scientists, what we need to do is figure out what we're going to build first, and then go into the toolbox and just pick the one that's going to give us the strongest argument, or is the sharpest tool to figure out what we want to do. That probably sounds cheesy, but I'm actually pretty passionate about it. I mean that. And when I say I wanted to ask Tor a question, my question about what you talked about is standardizing things. I usually preach not only in picking the tools, but to just try to get all of the stats and stuff out of mind when you're trying to figure out your paradigm and how you're going to do an experiment. And if that tool doesn't exist, go make it up. Or find some person like me down the hall and say, make this for me. Because there aren't a lot of standards, I think. I think a lot of what we have now is a toolbox is just people sitting around saying, man, if only I could do this in a multivariate way. Well, how would we do that? And then 10 years later. So I hope that answers your question a little bit. And then my question for Tor has been, do you worry that standardizing things might stifle some of the creativity in this kind of organic part of the scientific process?
Yeah, I'm all for a creative process. I love it. And I admire, when people come up with really new concepts and things, like some of these inter-subject correlation things is one example, and many others. But I guess I would say, you should sort of standardize the things that you're not trying to innovate on, and that actually lets you innovate on the things that you do want to innovate on. So, for example, like if you wanted to do something, super creative with MVPA, but you had to go back and program in your own brain normalization, right? Or whatever, segmentation, or what, you'd never get there, right? So that's why I think actually in some ways, standardizing things that can be sort of like done once and don't redo it, right? If it works, that gives you building blocks to be really creative. And if you think about the number, going back to sort of Larry's talk at the beginning of the day, first principles, getting really good signal, better, higher resolution signals from the brainstem and from other, cortical columns, et cetera. Though that just expands our toolbox. And so there's so many pieces that we just rely on to work, right? Sorry.
So I maybe want to bring up an issue that spans several of the talks, especially the last 4 talks of today. And that's sort of a common theme is that we all agree that prediction is very important, because out-of-sample prediction is a good way of really putting our models to the test. And when we can do that with actual data that we haven't had access to while we fitted the model, then that's really very compelling. But there's sort of a number of differences in the approaches that were presented. Some of the people presented within subject prediction, like Michael Pratt, for example, looked at decoding, and there we usually care about very fine-scale patterns that might not generalize between subjects, so we do within subject prediction. And some of the other talks used between subject prediction, partially with a similar goal of decoding, but to some extent also to relate brain activity patterns to disorders and group differences. How does this all come together? When should we be using spatial patterns versus functional connectivity or representational similarity? And when should we be doing within versus between subject generalization in our predictive analyses?
Okay. Well, so I think there's very different tools for different purposes, right? But if you focus on the goal of prediction as a goal, right? I would argue for the simplest model that works well enough. So if you think about within subject versus between subject, right? So I would say, you know, if you can come up with a between person, a population level model that works, it's got advantages. It's simple. You can interpret it better. You're finding something that is sort of holds across people, less likely to be influenced by artifacts and other sources of noise and so on. And if that does, you know, about as well as within person prediction, then you've learned something about the sort of nature of representation and its generalizability, right? On the other hand, you know, there are many cases where it's just not going to work. If you're trying to do ocular dominance columns, they don't think it should work, right? And in which case then, you know, you need to go to what I would call a more complex model, right? Because you have, you know, number of parameters times number of subjects, et cetera, right? And then I think you'd use a RSA, you know, representational similarity, for a totally different purpose, really, if you want to try to understand, you know, sort of the nature of what's being encoded across stimuli and so on, which is complementary.
So one difference seems to be that if we're interested in particular pieces of content, like particular stimuli and in particular brain regions, then usually it's difficult to generalize across subjects, and we need to do a within subject analysis, either encoding or representational similarity or decoding. But for some of these other applications, it seemed to work well to look at very coarse scale patterns and often whole brain patterns also, right? Which is another question. So these things also don't need to go together, right? We could take region of interest approaches, but still try to go across subjects. So I guess as a listener over the course of the day, I'm sort of looking for a framework of which is appropriate when, or to what kind of question, or what are the questions, basically, right? One question is what information is where in the brain, and that was addressed by Michael Prati very, very much. Chris Honey also did decoding of movie, where are we in the movie, and used functional connectivity fingerprints, but could also have used spatial patterns equally well. And I wonder if that would have worked equally or even better or not as well.
So I think that part of the reason the very coarse-grained patterns worked well is, so this gets into kind of more conceptual territory, but cross-brain decoding, you can decode even across perceiving part of a story and remembering part of a story, or a particular part of a movie, and you can do that across subjects. And if you can actually see the similarity of the patterns with your eye, right? So there was a paper from Janice Chen in Nature Neuroscience at the end of last year, and she has this figure where you can just look at a kind of snapshot of the brain state, for a particular part of a movie, and you can see it in this set of subjects, and then a replication group, and you can go to the next, 20 seconds later, and you can see this group, and then a replication group. So there's a certain sense at which the brain has both scales available to it. There's this very coarse-grained state that shifts usually on a slower time scale, and it may be that that's actually something that matters in the brain because of the way the world is organized. So the world has this kind of nested information structure, and as a result, coarse-grained states of the world, such as what you are currently doing and who is around you and so forth, they generally don't very, very rapidly. And so it might make sense for the brain to have a kind of a low-dimensional representation of those features, and that they're coarsely represented. But if you wanted to be doing something like decoding whether the particular narrative that we presented was presented in a sort of sarcastic tone of voice or not, then that's going to be more difficult at a between subject level, I think. So I guess it depends a bit on your question, which is the answer to a lot of these questions. But I certainly don't think it's the case that the information is only at one scale or the other. I think that it's quite possible that the large-scale patterns that we see are actually meaningful from a functional level. They're not just aggregates of the true fine-grained pattern. Yeah.
This might be a follow-up to that question, but it's really, this has been running on my head since your talk, Mike, is, yeah. Oh, sorry, yeah, well, okay. Well, I'll say it, and then we can go back to your question maybe, but it's just that decoding doesn't imply representation. And you showed us examples of how, like, if V1 can decode sounds, it's not that we think that V1 represents sound, identity as an object. We could talk about that, but that's something that's, I think, a sticking point for me.
Yeah.
Wait, I'm a little bit confused about this comment. If V1 can decode sounds, there is information in V1 about sounds. I don't know what you mean by representation, but if V1 decodes sounds, there is information about sounds.
Right. So the way I look at it, I roughly agree with Tor that when the information is present, it isn't by itself sufficient to justify a representational interpretation. A representational interpretation implies that the information is not only there, but serves the function in the context of brain information processing to convey that information. And so ideally, you'd have some additional evidence that other regions are reading that out, or that it's related to behavior or something. Of course, we have these problems with all methods that we always are trying to understand a network, and the interpretation we put on it is always somewhat speculative, right? So I think in many cases in the visual system, it's very compelling to say that it serves the purpose of a representation, and I wouldn't be too harsh on people, making that interpretational step, but it is adding something to it.
And just to follow up, I think if you removed someone's visual cortex, they could still hear a duck quack, but there's a subtle argument to say that my representation would be richer if I could imagine that duck quack, and whether that is part of the hearing the quack or not is, I know it's for philosophers.
Hello. Thank you. Thank you for the excellent talks through the day. Actually, I think my question takes the whole topics that we heard from you today. As a psychiatrist, my main angst and my main drive to be here has to do with how can we apply this to a clinical setting. So to end up my talk, my question is, do you think even if eventually all the field has everything standardized and let's say everybody agrees on a standard of preprocessing and everything, don't you think, I want to hear your thoughts on the sample size. Don't you think that in the end, after all, we'll also need a large sample size, as we've seen with genetic studies, for example?
Yeah, the sample size question is a tough one because you don't really even, we're at such a early stage in this work that we don't really even know what effect size we're searching for. We don't have a very clear sense of what form the right answer is going to take in the end. I think that work that's already been published by several groups, a half dozen or more, shows that you can do meaningful, interesting things with not crazy sample sizes, like in the hundreds, in terms of individual predictions, if that's what you're asking about, like predictions that might be clinically meaningful in a real life setting. But you bring up a great point about genetics. We heard about that in the morning a little. You're probably going to need much larger samples to learn about the genetic underpinnings of most of these mental illnesses, and that's probably going to mean that it's not practical to get fMRI on all of those folks, but there might be some means of bridging the two, and I think we're a ways off from that, but that isn't necessarily an insurmountable obstacle. That's my view on it.
All right, we have five minutes over, so we should probably close it at this point. Let's thank all the speakers again. And thanks to you for coming.